Hello everybody. Today we will gonna discuss about the KEGG database, and this database is in Japanese database which will give us a result about like these. These results are regarding to primary, secondary metabolites, the amino acid, and many more things like disease and much more things. So we will proceed to the data. First of all, you must watch a full video in order to learn about this cake. So for moving to these data, we will proceed to the CAS cake. This is a database which is related to the cake database, and uh, which gave us a full explanation and a full blast with the cake database. So whenever we open it, so this is another database. We must look forward to the request for the job for the KEGG. So we are doing a blast of our data with the KEGG. So first of all, we must select our data. If we have a smaller data, so we can paste it here like in faster sequence. And if we have a larger data, so that larger data is sometime in Swissprot data or sometime in pasta, but these data are related to the RNA sequence data, and the other the RAD sequence and the cDNA. So first of all, we must choose our file from here. For example, I have a file like um, we will proceed to this file. So we are here about the Swissprot data. So here we have the Swissprot data. These are the Swissprot data. Like for example, we have a Swissprot data of Artemisia species. So what I'm doing is uploading that data. Like I uploaded it here, and here there is uploaded. If we have a text data, so we will upload here. If we have file, we have uploaded. But our data are RNA sequence data, so we can't say that these data are nucleotide, so we cannot take these. So we will give a name for it, like for example I'm giving it Artemisia. So we will give an email, but that normal email like which I have are not mostly acceptable. We must look forward for institution email. Like for example, we have a Harvard email, like Harvard, like many emails which are institution which are related to the institute. So we are doing it for eukaryotes, so we will select, and we can also choose our organism manually. Like for example, if I want, so I can uh, blast my data about the measure species with these data. Like for example, I choose these species. So if you look at here, these species are selecting by themselves in these bars. So maximum you can only upload or blast your data with 19 species. So whenever you upload these species, so then you have to blast your data and click on compute. So these result will give you in 2 to 3 days and sometime in 1 day. But uh, I don't have any time to wait till now. So whenever they give you result, so the result will be in PDF file in your mail. So whenever you open it, so the result will be that time. So if you look to the data, so these are our sequences or short form or our sequences. Like for example, the Artemisia species has these sequences, while it has been blasted and show approximately 70 to 90% coverage with the KEGG database data, and that is here. So these information about regarding the coverage of our data, and you can give these coverage depending upon your data need and your need. which give them mostly infiltration process before this is Sprott data. So if we uploaded the data here, so it will be okay because the Notepad++ is in software which is acceptable for many data, while X is not a supportive format for these data. So we will copy these data. It's large data, so in Excel it's very hard. So first of all we upload here to make sure our computer will be not hacked. I'm selected my data. So the first one was our data, while this is in KEGG, and the KEGG gave us a result about the KEGG KO, which means to a KEGG orthology. So we have selected our data. Now let's note this. I also copied that data, but let's note this. It's 002467. So we will search it here for, like, we have 002467. Now we are pasting our data here. So it's large data. So what are we doing? Moving it for sort to make sure our data is easier for filtration. There are many duplicates, so we will also remove the duplicates from these data. Here it is. Press Ctrl and Shift and then click on the top. So now our data is copied. We will paste it to another section, remove the duplication. So we will expand it just for B column. So there are 3607 unities which show coverage with our database. So now we are moving to the KEGG, which are our main aim to proceed. So the KEGG database is here. We have a KEGG database, and these database giving explanation about the pathways, the KEGG orthology, the gene, the bright about anything related to these genes and chemical disease, drug, and many other medicines. So we will proceed to the KEGG orthology because the data we have, these are the KEGG orthology, which means that KO KEGG orthology. So we will paste our data here, like I am copying this table and paste it in here. I pasted my data, but because these bars aren't so large, so the data can't be expressed here, but it is uploaded. So we will click on map chart because we want a map chart for our data. So here we have a full result regarding our data. So there are metabolic pathways which show coverage of 958 unigenes with our data. And there are many other explanations, many pathways like secondary metabolites and the microbial diverse environments are many pathways, and these pathways can be represented with these ID specific, but these are not KEGG orthology. It has a different meaning. Like for example, we are selecting the secondary metabolite biosynthesis. So here they give us a full explanation about these KO IDs, these 537, which are involved in the secondary metabolite biosynthesis. And as we know that these secondary metabolite are much applicable in our community. Like for example, they are applicable in druggable compounds. So we are click on that side of the secondary metabolite. So here we have in new tab, so they gave us a full explanation. We can also download these data. Like for example, I downloading this network in image file. So they will give us explanation about that. And if we look here to the other side, so there are different modules regarding this pathway. Like for example, I am clicking on these pathway, these modules, so they are giving us explanation. So if we move here, there are different like so if we see there are red color and the blue, the red color that show the enrichment of our data with the KEGG database. Like for example, I click one of the module which show full enrichment. Like we can say that if we have these data like threonine and click on to be make sure it is safe for the other type. So we have this data, so it is always represented by another graph. So we are clicking it at a new tab, opening it a new tab. So here we have a threonine, and they gave us a full explanation about that is a primary compound, and these are the main genes which are involved in these biosynthesis, and these are other. So the last product is threonine. For some time we can express them in secondary metabolites, if am I not wrong. So we will look for so to make sure in secondary metabolite we have different classes like flavonoids, alkaloids and terpenoids. So we will proceed here first of all to the flavonoids. So it's flavonoids. So here we have flavonoid biosynthetic pathway, and they give us a full explanation about all those compounds which are present in the flavonoids. Like for example, we have different compounds and different enzymes. So these some are blue in color and some are red in color. So we can say that the red color show an enrichment with our plant data, like we have that. So it is a biosynthetic pathway for naringenin chalcone, and the other is naringenin. So if we move here to look forward, if we, our main product we want is a naringenin, so we click on that. So if we look backward to the pathway, so there are two main enzymes involved in these pathways. Like for example, one is that and the other one is that. So the KEGG is giving much more explanation about these pathway. So our compound which are we aiming is that naringenin, and they also were giving us a different line like pumpkin, and the most important is human databases and the knapsack. So this gave us a full explanation and also shows us that how many genes are involved in these pathways. So we will move here like the total gene which are involved in this database isn't 27, but it isn't clear here. If you downloading the RDF file, so that will be clear there. So the total gene involved in these pathways are 27. So we are opening and proceed to our aim. So that was the naringenin and the two main enzymes which are these in pathway. So that was the chalcone synthase, and the other one was chalcone isomerase. So here we have a chalcone synthase and chalcone isomerase. So they give us a full explanation on, and also give us more explanation about these enzymes and the compound. And most important, if we don't have these enzymes, so there are many orthology for that which are also available here, like we have here. And these all shows that the most important gene which are responsible for biosynthesis of these are shown here. So we are moving to next point. So this is about the secondary metabolites and primary. So now we are moving to the enrichment analysis. So if we see here, so the total enrichment in this database of our plant is 18. So we can say that like in KEGG database there are total 18 unigene which show coverage with our data. So we have here we will edit our data. Like for example, I already done that before. Like flavonoid biosynthesis, this is in pathway, and this is giving us a unigene, that the total pathway in these flavonoid are 27, while the which show coverage is 18. So we can also proceed all of our data to here from there. Like for example, we have different pathways and there are different genes involved here, so we can make a better charts from these. So we are here, so now we are moving to that how to predict and compound from these pathways. So we will move here, like for example we have already start working on one compound, that was the naringenin, and the two main enzyme involved in these pathways are these two. So what we will have to do to click in naringenin, copy that, so that is the naringenin, and the two enzymes which are mainly involved is the chalcone isomerase. We will copy here and paste it into here for further processes, and the other one is chalcone synthase. So this is giving us a better explanation about the pathways. So here we have in secondary metabolite, we can say that in naringenin, and the two main enzyme involved in these pathways are chalcone isomerase and chalcone synthase. And if we look here, so the naringenin is a secondary metabolite, so definitely it will have medicinal purposes. Like if we see here, like medicinal purposes, so here we have naringenin is one of the major citrus flavonoids. So they also give information that these naringenin are anti-obesity, anti-diabetic, and anti-fibrotic. So congratulations, you are non-scientist because you predicted and compound. Like for example, these naringenin are till now never predicted in the species. Like we have the artemisia, and you told so that these compound in enzyme pathway is present. So congratulations. So never forget this step because we will moving into the next class. So that was the metaboanalyst, and metaboanalyst we will look forward for these KEGG IDs and these number which we have. Like every compound has their own specific number. Like for example, the liquid oxygen has this number, so this is a compound CID or compound ID. So save these and keep these in your mind because we will move further from here. So that was enough for today. Have a nice day, and also subscribe my channel and forward and share it to your friends.